This is part 2 of 3, looking at chapter 6 on energy, enzymes, and metabolism. The first part, we talked about energy, and we left off with a really brief introduction to the different types of catalysts. So we had the enzymes and the ribozymes, and these catalysts, they help to speed up reactions. We're going to be focusing on enzymes, which are those proteins that are used to help speed up reactions. So we're going to look and see how these enzymes actually work. Enzymes, what they do is they work on something called the activation energy. So if we go back and we just think about any type of chemical reaction, we need to put in a little initial input of energy to get everything started. And this initial input of energy is called the activation energy, or the energy of activation. And this energy, it does two things. It allows these molecules to get close enough together to cause bonds to rearrange. And then once these molecules are close enough together, then we can get our molecules to achieve what's called a transition state, where the bonds are stretched. And then eventually those bonds will break and you'll have new bonds that form. So this activation energy, it's really important for any type of chemical reaction. And the activation energy, here we have kind of a little diagram showing the activation energy. So if you think about, we have our starting material A, which is the rock down at the bottom. We have a person that has to input a little bit of energy to get over this little hill. And then once you get over the top of the hill, that rock will roll down to the bottom of the hill, and you'll end up with your products, which is labeled B in this diagram. So there are two ways to overcome this activation energy. So the two most common ways are adding a really large amount of heat. And this works really well in the chemistry lab, where we have, our Bunsen burners or our hot plates. We can add a lot of heat to get reactions going, to get them to go faster. But in cells, that's not a good option. We can't add a large amount of heat to our cells. So what cells or living things do is that they use enzymes to lower our activation energy. So you remember, enzymes are those proteins that are going to help to speed up all of these chemical reactions. So if we lower our activation energy, now instead of needing a large amount of energy, we just need a really small amount of energy. So we can add a really small amount of heat, and that will get those reactants to the transition state so that we can start our reaction. So we're going to be using this method number 2, using these enzymes to lower activation energy so we can get reactions started faster and we can get them going faster. So just to kind of present this idea in another form, so if you look at the graph here, on our x, or the bottom axis going across, this is the progress of our reaction. So we have our reactants on the left, and we want to get to our products on the right hand side. The y-axis, the up and down one, that's our free energy, or G. So remember, if G is positive, that means you have to add that energy. If the G is negative or below the middle line, that means that the reaction is going to release that energy. So we have our reactants over on the left, and this red line, it demonstrates that we need to add 2 amounts of energy to get up our little hill. Once we get those 2 units of energy added, we get to our transition state at the top of the hill, and then our molecules can very quickly go down the hill to form our products on the right hand side. So this is showing how much activation energy we need to put into the reaction without an enzyme. So if we add an enzyme into this, we're going to end up with this blue line on our graph. So now we start off with our reactants. If we look at the blue line, we see we have to go up a much smaller hill. In this case, it is half the height, or we need to put in half the amount of energy to get a reaction to our transition state. Once we get to the top of that little hill, we get to our transition state for our molecules, then those molecules can proceed on to produce the products over here on the right-hand side. So you can see that in this example, we cut the amount of activation energy in half by using an enzyme, which is really good because cells want to conserve energy, and we want to not have to add a bunch of heat to the reaction to get it going. So this is how these enzymes actually lower this activation energy. They lower this little hill that we have to get over to get things started. So one really big purpose for enzymes is to lower the activation energy that we just looked at. In addition, enzymes help to strain the bonds in your reactants to make it easier to get to that transition state. And then enzymes, they also position our reactants together to facilitate bonding or to help with the new bonding. And then in addition, enzymes can change the local environment so they can actually participate with this reaction, and you can have what are called temporary bonding that can occur between your enzyme and your reactants. So we're going to look at some of the features of enzymes. So here, enzyme is the pink molecule, and we're going to be having ATP interacting with glucose. So those are our reactants, or we're going to call them the substrates. So these substrates, the ATP and the glucose on our left-hand side, they're going to fit into a part of the enzyme called the active site. And this active site, this is where your substrates are going to lock into your enzyme. Once your substrates enter into the active site, and you can see they fit in really well into your active site, then we're going to have our enzyme substrate complex. So it's just our enzyme and our substrates together, and they're ready to start the reaction. Our reaction proceeds. So here what's going to happen is our ATP, we're going to take one of the phosphates off the ATP and put it onto our little glucose molecule. And you can see that in the middle picture of our enzyme. So our yellow phosphate moved from the ATP onto the glucose molecule. So once our reaction occurs, then our enzyme will release the products, and we end up with ADP, so adenosine diphosphate. It has two phosphates attached. And then we have a glucose phosphate, so we have a glucose molecule that has our third phosphate attached onto it right there. So those are the products that are produced. So I mentioned that our substrates are starting molecules. They bind into the active site on the enzyme. So enzymes, they have a really high affinity or a degree of specificity for certain substrates. So enzymes are very, very specific for what type of reaction they will do. And this is due to these substrates are going to fit into the active site like a lock and key. So only certain substrates can fit into the active site. Or you can think of it as like puzzle pieces going together. Only certain puzzle pieces will fit together. Once your substrate gets into the active site, some enzymes will also have what's called an induced fit model or induced fit interaction. And this is basically the enzyme is going to wrap around the substrate and kind of hug it and keep it in place. So induced fit, it changes the fit a little bit so that your substrates are close together and that they can go through the reaction. So we talked about enzymes help out with reactions, and enzymes are not used up in these reactions. So sometimes the cell wants to inhibit or stop a reaction from occurring. So inhibit means to slow down the reaction, or we can actually stop a reaction. So there's two ways that cells can inhibit or stop a reaction. One is called competitive inhibition. This is where we have a molecule called an inhibitor. If you look at the bottom, it's going to be the bright pink molecule is our inhibitor. The blue molecule is our substrate. So that's what the enzyme actually works on, is the substrate. So during competitive inhibition, we're going to have an inhibitor. It's going to compete with the substrate to bind into the active site. If your substrate gets into the active site, the reaction proceeds. If the inhibitor enters into the active site, it's going to inhibit or stop or slow down that reaction. So the inhibitor inhibits the ability of the substrate to actually get in to the active site. So competitive, just remember the inhibitor and substrate, they're competing for the same site on the enzyme, the same active site. Another type of inhibition is called non-competitive inhibition. So instead of the inhibitor and substrate competing for the active site, our inhibitor, which is now the circle pink molecule, it's going to bind onto a secondary site called an allosteric site. So this is not the active site, it's a separate site on your enzyme. So if this inhibitor gets into the allosteric site, it's going to cause the enzyme to change shape. Specifically, it's going to cause the active site on the enzyme to change shape. If the active site changes shape, it's no longer going to fit with our substrate over here. So if our substrate can't get into the active site, it's going to turn or stop that reaction from happening. So again, this is non-competitive. Our inhibitor is going to bind on to a different site on your enzyme, and then it will cause the enzyme to change shape. If the enzyme changes shape, it can no longer do that reaction. It can't work on that substrate. Some enzymes require helpers in order to do reactions. So we have prosthetic groups. These are small molecules that are permanently attached to the enzyme, and they help the enzyme speed up reactions or lower that activation energy. Some of these groups are called cofactors. These are usually inorganic ions, usually the metals. And these inorganic ions, they're going to temporarily bind with the enzyme and help with the reaction. And an interesting thing about cofactors are the minerals that we are encouraged to eat in our diet. So these minerals, the reason why we need these minerals, so things like iron or zinc or magnesium, we need these minerals because they are cofactors. They bind down with certain enzymes that help our cells do certain reactions. So it's good to eat your minerals. Coenzymes, another type of requirement for enzymes. These are organic molecules. They also help the enzymes, so they participate in the reaction. And these enzymes are left unchanged, so they can be reused. These coenzymes can be reused. Coenzymes, these are going to be your vitamins. So vitamins are organic molecules that are going to help with these different chemical reactions. So when your parents tell you to eat your vitamins and minerals, now you actually know why vitamins and minerals are important in your cells. They're going to help with the enzyme to help speed up all these different reactions. So besides enzymes helping to speed up all the different reactions in your cells, and the neat thing of having enzymes relate to your vitamins and minerals that you should be eating in your diet, the coolest thing about enzymes, or at least in my opinion, the coolest thing about enzymes is that they really depend on the type of environment that they are in. So most enzymes, they function maximally or at their best in a really narrow range of temperature and a narrow range of pHs. And this is a lab that we're going to be doing in the labs for the enzyme lab. So you're going to be taking an enzyme and putting it at different temperatures and pHs to figure out where that enzyme works the best. Outside of these really narrow ranges, the enzyme function is going to decrease. And it's either going to be because the molecules aren't moving fast enough, or the enzyme, because it's a protein, the enzyme can denature, or basically it's going to fall apart. So here we have an example of denaturing. So over on the left-hand side, we have an enzyme that's in a native state. So it's folded together in its 3D shape. It has a really nice active site. If you heat this enzyme up to almost boiling, it's going to cause all of the different bonds in the enzyme to break. So the enzyme's going to said to be denatured, and we're going to end up with an enzyme that is basically falling apart, and it will no longer have its active site. And remember, we really need that active site for the substrate to get in there so that we can have our reaction. Okay, so this is just an example of how we can actually measure enzyme function or the rate of a chemical reaction at different temperatures. So in this example, we changed one factor in the environment, the temperature. So we have from 0 degrees Celsius, or like about freezing, up to between 50 and 60 degrees Celsius. And then we measure the rate of our chemical reaction, which is a proxy or a measure of how we can measure enzyme function. So the higher the chemical reaction, the better the enzyme works. The lower the rate of the chemical reaction, the lower or the worse the enzyme works. So as you can see from this graph, our highest rate of chemical reaction, or the highest, best temperature that the enzyme will work at, is about 37, so between 35 and 40 degrees Celsius. And 37 degrees Celsius is an important temperature because that is human body temperature. So this enzyme works best at human body temperature. So again, we'll be looking at this in lab, looking at different temperatures and different pHs and how it relates to enzyme function. So this second part of Chapter 6 really looked at enzymes and some important parts of how enzymes function, and then enzymes need cofactors or other molecules to help them function, and then how enzymes actually can interact with their environment.